Question 8. A scientist wants to purify a recombinant protein containing a histidine tag from a bacterial lysate. Which technique would be most appropriate? Okay, so let's think about this. A researcher is making a protein. Basically, a recombinant protein is one that's been made and engineered by a researcher. And they're adding a histidine tag, which is often the case when you're trying to purify a protein from a large sample, like a bacterial lysate. You add some sort of tag that you can specifically look for when you're trying to isolate your protein, so you know that only your protein of interest is what was isolated. And so again, let's just think about this. We have some sort of protein, and on the end of it, we add a histidine tag, which is usually 6 histidine molecules, all strung together. And so what technique would be most appropriate if we're trying to isolate proteins just with this histidine tag? Well, let's go through the options. A. Ion exchange chromatography, because histidine tags eliminate charge difference. Well, what does that even mean? Eliminate a charge difference? Adding a histidine tag is not going to appreciably change the charge of an entire protein. Think about it. This tag is 6 amino acids long. Our protein could be 100,000 amino acids long. Adding 6 histidines is not going to appreciably change the charge of the peptide. And so ion exchange chromatography would be useful if we have an extremely negatively or positively charged sample. But that's not the case. We're talking about a histidine tag. B. Size exclusion chromatography, because histidine increases molecular size. Well, remember, we're using a histidine tag because we want our method of purification to be selective for the histidine tag. So yes, will adding a histidine tag increase molecular size? Technically, yes, but it won't do so in an appreciable way. Remember, we're only adding 6 histidines. If you have a peptide that's hundreds of thousands of amino acids long, adding 6 histidines isn't really going to change the molecular size in any appreciable way. And again, we're trying to separate it from a bacterial lysate. So there's going to be plenty of proteins that have similar size to our protein of interest. And so we're not taking advantage of the histidine tag as something that can be selected for specifically, because our protein of interest, as well as hundreds of other peptides, all of similar size, are all going to elute together or get stuck together in the column. And so this method doesn't take advantage of the histidine, which is what the question is really trying to get at. Answer choice C, SDS-PAGE, because proteins with histidine migrate differently. Well, that's just not the case. Remember, SDS-PAGE takes advantage of strictly a protein size. And there's going to be hundreds of thousands, again, of proteins with similar size. And so C is not correct. And then D, affinity chromatography using a nickel-containing resin. And that's correct. Remember, nickel is a selective way to bind for histidine. And so if you have nickel in a resin, that means any protein that floats by with the histidine tag is going to bind to it strongly. And so you can control for this by letting all proteins get flushed out of the cell, and then only proteins with histidine are bound to the nickel. And then in a controlled manner, you can decide when to interrupt this interaction, and then all you have left in the column is just your proteins bound to histidine. And so that's why this method is selective and takes advantage of the histidine tag to specifically isolate your protein of interest. Question 9. A protein mutation decreases the number of hydrophobic residues buried within its core. So we're decreasing the number of hydrophobic residues in the core of a protein. The mutant protein forms insoluble aggregates. Which explanation is most likely? A. Hydrophobic residues normally stabilize proteins by interacting directly with water. Well, that's not the case. Remember, hydrophobic residues like to be inside the core of a protein. They do not want to interact with water because that decreases the entropy, since water has to form hydration shells around hydrophobic residues. B. Increased hydrophobic residues increase solvent entropy and cause precipitation. Well, this isn't going to be the correct answer either. Remember, increasing hydrophobic residues is going to decrease solvent entropy. All those waters that were previously free to move around now have to form an ordered hydration shell around hydrophobic residues. And this is going to decrease the entropy of the solution. And so that is not what's going to be the cause of precipitation. C. Loss of hydrophobic interactions prevents peptide bond formation. That's not the case. Remember, hydrophobic interactions is for secondary, tertiary, quaternary structure of proteins. It has no effect on primary structure, and peptide bonds are primary structure. So that answer choice doesn't make sense. It's not correct. D. Improper folding exposes hydrophobic regions, causing protein-protein interactions and aggregation. So that makes sense. Let's think about it. Pretend we have a protein with a hydrophobic core, which I'll just draw as this jagged structure. And so let's just pretend our protein is this circle. Let's say the mutation cuts this protein in half, right here. So now we have one half that's like this, and one half that's like this. We know that since we're in solution, this half, which was previously exposed to water, is probably going to be polar and is probably going to love water molecules around it. But this part that was previously in the interior of the protein does not want water molecules to be binding with it. It's probably non-polar. It wants to be bound to other hydrophobic components. And so if we have a bunch of these floating around in solution, what's going to happen? Well, we know that these specific areas don't want to be bound to water. So where are they going to bind to? Well, they're going to go back to binding to each other. And what you're going to end up with is protein aggregates. So all these nonpolar regions are going to be binding to each other to inhibit binding of water. And that's why you're going to get insoluble aggregates. And that's why D is the correct answer choice. And finally, question 10. A protein mixture is separated using several techniques. Protein X has a pi of 5 and a molecular weight of 100 kilodaltons. Protein Y has a pi of 9 and a molecular weight of 20 kilodaltons. The mixture is placed in a buffer at pH 7 and analyzed by SDS-PAGE. Which statement is correct? Okay, so we're analyzing by SDS-PAGE and we're trying to separate protein X from protein Y. And so let's look at which statement could be correct. A. Protein X migrates further because it is negatively charged. So we know that's the case. We know that since the pH of 7 is greater than the pi of X, which is 5, we know that X will be negatively charged. However, the migration of X is not going to be affected by a negative charge. And remember, that's because we're using an SDS-PAGE. And so this creates a uniform negative charge on all proteins. And so it doesn't matter if X is going to be negatively charged because it's pH versus pi. It doesn't matter because all proteins, X and Y, are going to be negatively charged. And so that's why A is incorrect. B. Protein Y migrates further because it has a lower molecular weight. Okay, well that's plausible. We know that SDS-PAGE separates proteins based on size. And we know that proteins that are smaller will migrate further. So that makes sense. C. Protein X migrates further because it has a higher molecular weight. Well, this is the opposite. Again, a higher molecular weight would mean a protein migrates more slowly because it has to navigate through all those pores in the gel, and a larger protein will have a more difficult time navigating through smaller holes. And then D. Both proteins migrate according to their pi values. Well, that's not correct. Remember, this question is testing to see whether you understand what an SDS-PAGE really is, and whether you understand that it makes uniform negative charges on all proteins. And so this is a way that the MCAT might try to trick you. It gives you PIs. It gets you wanting to think about PI versus pH and how a protein is charged. But because we're using an SDS, that has no impact on how proteins will migrate. And so that's why the correct answer choice for this question is B. And there it is. Those are the 10 questions I had prepared for today. I hope you guys were able to get something out of this video, and please feel free to leave any questions in the comments, or even if you have any material that you are studying that you are having difficulty with and you want covered in future videos, please feel free to leave that in the comments below, and I wish you good luck with all your studying.